All right, so we're going to continue on with Unit 2, Section 5, which is going to deal with Learning Target 10, and this is going to focus on enzymes. So again, a review, a chemical reaction are reactions that are building new chemical bonds, okay? And remember, in those chemical bonds, energy is stored, so energy has to be put into substances for them to react, and that energy that is put into it is stored in those chemical bonds, okay? And a lot of time with chemical reactions, you are creating something new in comparison to what you started with. All right, also keep that in mind. All right, so an example, when glucose molecule is created through photosynthesis, energy from the sun is being captured and stored in those chemical bonds. And then in cellular respiration, we'll break down those glucose molecules, and the energy from the sun that is stored in the phosphate bond, the phosphate bond between the second and third phosphate group. When that bond is broken, that energy will be released. And that also plays into that kinetic and potential energy. Potential energy is stored in the bond, kinetic energy is released. All that should be reviewed, and that should all be ringing a bell. All right, so activation energy. It's a definition here. Activation energy is the energy that is required to start or begin a chemical reaction. Okay, so again, all chemical reactions require a certain amount of energy to get the ball rolling, so to speak. This is known as the activation energy. Some chemical reactions store energy. This means that they end with more energy than they begin with. These reactions tend to require more activation energy than others. So if we look at this graph, let me get my pointer. If we look at this graph, then we can tell that this is a reaction that stores energy. This is where we start here on the left-hand side, and this is where we end. Okay, so the graph's line is higher at the products than it is at the reactant. So that tells us that this particular chemical reaction is storing energy in bonds. We're not breaking things down, we're making bonds here. In comparison, some reactions end with less energy than they start with. And these reactions tend to not require as much energy to get started. And once they get started, it happens very quickly. How do we know we're losing energy here or we're releasing energy? Where we start is always higher than where we finish. Okay? The energy at the reactant's point of the graph is higher than the energy at the product's point of the graph. An example of this is combustion of gasoline. Okay, doesn't take a whole lot of energy to get that started, and once it gets started, it goes pretty quick. All right, so what's a catalyst? We're talking about enzymes, so a great example of an enzyme is a catalyst. This is something that speeds up a chemical reaction by lowering its activation energy. So think about the vocabulary here. Okay, activation energy is the energy required to start a chemical reaction. So a catalyst can lower that. So meaning that a catalyst can not only make a reaction happen faster, it can also make it happen more efficiently. Okay, and that's the name of the game in nature. You want to be as efficient and as quick as possible. All living things in their body systems want to achieve this. A catalyst helps us make that happen, helps animals make it happen, helps plants make it happen, it helps nature make it happen. So enzymes are biological catalysts. These molecules speed up reactions in organisms' systems, conserving energy and time, making them a more efficient organism, which is going to ultimately mean higher survival rate. There are almost always proteins, enzymes are, and they usually end with the suffix ase. For example, okay, the one that we'll reference a lot in this class is salivary amylase. This is an enzyme in the saliva that breaks down the bonds of starch. It converts them into individual glucose molecules. Starch is very hard on our digestive system to digest. Okay, I'm not going to get into the specifics, but it's not very easy to digest. So we have an enzyme, a protein in our saliva that begins the digestion process of complex starches as soon as it enters our mouth, as soon as the saliva comes into contact with it. Our bodies can break down glucose a lot better than starch. Okay, so you break starches down into their monomers and you have glucose. This is a chemical reaction. This is a chemical reaction. All right, so the enzyme substrate complex. Let me make sure I didn't skip a slide. I did not. Okay. Enzyme substrate complex. This is the combination of the enzyme and its substrate. A substrate is the reactants in the enzyme reactions. This is the molecule that is being changed by the enzyme. So with salivary amylase is the enzyme and the substrate is the starch. The starch is being changed into glucose. Okay? So the enzyme substrate complex simply just shows how the enzyme is going to attach to the substrate, which will eventually change the substrate, but the enzyme will stay intact so it can do the job over again. In enzyme reactions, the substrate changes, but the enzyme remains unchanged, which allows it to be able to perform more reactions. The enzyme and the substrate combine on the enzyme at the active site. Each enzyme is specifically designed for its substrate. Remember, with proteins, the shape dictates the function, and the shape of the enzyme dictates which substrate it will react with. That's important, okay? The shape of the protein gives it its job. You need to understand that. So its job or the enzyme's shape is based off its protein shape. And the shape of the enzyme is going to tell it what type of substrate it's going to attach to. For example, salivary amylase will only attach to starch. It's a particular shape at the particular active site. It will not attach to anything else. It is specifically designed to attach that one type of macromolecule. Enzymes can either combine two substrates to create a single bond or split a single reactant into two products. Okay, again, salivary amylase is splitting starch into individual units of glucose. On the other side of that, starch synthase, there's the suffix ase, A-S-E, synthase, is a molecule that combines units of glucose into chains of them called starch. And I believe this will be the last slide. Enzyme reactions can be regulated by the body. This makes sense if you think about it. One of the ways that it is regulated is by the amount of the substrate. When the substrate is gone, the enzymes will no longer react. And this also means the body will no longer make those enzymes to do a job. If there's no starch to be broken down, then why would the body waste energy to make salivary amylase? Doesn't make sense, right? It's a waste of energy. So how does it get rid of enzymes? Well, the body denatures them. Okay, when you get a low-grade fever, you may be denaturing enzymes, getting rid of excess enzymes that your body doesn't need right now. So temperature and high and low pH will denature enzymes, as will any other type of protein. And I believe that is the end of this particular presentation. Be sure you complete the form that goes with this, and if you have questions, let me know. Thank you.